a very interesting paper by Dr. Barnden used MR, but in a totally different way than MR has been used in studying chronic fatigue syndrome. Past studies have really been studies purely of anatomy. Is there anything different on MR? Are there any little white spots suggesting demyelination in white matter? Are there any gray matter volume differences in MR or ventricular size differences? Here, what they did is break down, as I understood it, break down MR into the 200,000 tiny little components, voxels, that make up the entire human brain. And then for each voxel, or maybe it was for each tightly linked group of voxels, tried to correlate the intensity of the signal from that voxel with the symptoms that the patients were reporting, to see not only if there was a correlation between MR signal intensity in general and chronic fatigue syndrome, but whether if there was a correlation between certain symptoms and certain parts of the brain where you would expect to be seeing the action with regard to those symptoms. And they found that there were multiple locations in the brain where changes in MR images were associated with CFS severity scores or surrogates for severity scores, and that many of the locations where these signal reductions typically were found were consistent with the symptoms that were being expressed by the patients as a group. The abnormalities of the medulla and insula were consistent with the involvement of the autonomic nervous system that most, but not all, papers have shown in CFS. This involvement of the cerebellum with coordination of motor functions, I would say that's a little softer in terms of this report by patients with CFS, and the prefrontal areas where the correlation between both impaired motor function and fatigue made sense. So a very interesting way of using MR to look at CFS that, in my knowledge, had not been tried before. Dr. Mike also, I think it was he, yes, I think it was, talked about a characteristic abnormality in some, but not all, patients with CFS on the P300 wave, and another electroencephalographic abnormality. Cytokines as mediators of symptoms. Dr. Klimas's group in a study, again, today you can simultaneously measure cytokines, either message for cytokines or the cytokines themselves, much more easily and less expensively than was possible a decade ago. And Dr. Klimas found that a lot of pro-inflammatory cytokines were measurable at increased levels. Surprisingly, one that prior studies from Dr. Klimas's lab and others have found was elevated was not in this study, but a whole set of pro-inflammatory cytokines were. The argument has been raised whether these elevations are high enough to actually exert effects physiologically on brain, particularly to create in brain some of the symptoms of this illness. But there seems no doubt that there is a difference, a measurable difference, the physiologic importance of which remains to be identified. And probably the way to do that is with cytokine blocking drugs in controlled trials. Other cytokine findings by Dr. Klimas, or I would summarize here as I understood them, that basically this study confirmed what many, but not all, studies have shown, which is the Th2 cytokines there is a shift in the direction of Th2 cytokines, and away, therefore, from Th1 cytokines. And that IL-8, which is important in natural killer cell motility, is 42% lower, which could be part of the explanation of the functional impairment in NK cells that Dr. Klimas's group, our colleagues at Dana-Farber, and others have identified. Dr. Morovska's study also looked at cytokines, particularly these 3 cytokines in serum samples from CFS patients. I put these red arrows in to focus you on the normal subjects, that is, those who did not have evidence of an acute viral infection with HHV6 or 7 versus those that did, and the TNF-alpha levels and the IL-6 and IL-4 levels were higher. Cytokine profiles may depend on clinical subgroups. Dr. Porter, and this I think was sort of a harbinger of Andrew's comments this morning, pointed out that in their studies in Chicago, the patients who clinically reported a viral onset tended actually to have more of a Th1 pattern, whereas those with a non-viral onset tended to have a Th2 pattern. So that suggests that a post-infectious fatigue syndrome may not be representative, at least, of all cases of chronic fatigue syndrome. My memory of Dr. Porter's talk is that about 50% of patients fell into the viral onset group and the other 50% the other group. Th17-related cytokine abnormalities were reported by Dr. Demelier and Dr. Fremont. This is very interesting because Th17 response and IL-17 specifically and the related pathway genes are a really hot new area of immunology, very important in autoimmunity and probably in a number of gut-related diseases, since the system is important in maintaining the integrity against infection of the gut mucosa. Doctors Fremont and Demelier had previously reported that a particular polymorphism of the IL-17F gene, characterized by this His-Arg substitution, that confers protection against inflammation, that this polymorphism was underrepresented in patients with CFS, which would lead to increased inflammation. At this meeting, they also reported tentative data that a polymorphism in the gene for IL-6, a pro-inflammatory cytokine, a polymorphism that causes increased production of this pro-inflammatory cytokine was more frequent in patients with CFS. So 2 different polymorphisms that could increase the inflammatory response and could explain some of the symptoms of this illness, theoretically. I think I've just said that. Dr. Petty, in a remarkably well-done presentation this morning, my model of a presentation: tell them what you're going to tell them why it's important, tell them clearly about your methods, tell them about your results in a way that's comprehensible, and what your cautious inferences were, and that was a model. MicroRNAs, as he summarized this morning, for some of you may not know, are arguably the most important event in biology of the last 25 years. The central dogma of molecular biology, first promulgated about 60 years ago, has only been amended a few times, and this is one of the times. We now know that our genome not only transcribes full-length genes into messenger RNA, but also transcribes little segments of DNA into tiny little microRNAs. And these microRNAs dramatically affect the expression of genes. In most instances, at least studied so far, they suppress the expression of a gene. In some cases, they seem to augment it. But gene expression is really important. It explains biology. It explains why the cells in one tissue are so different from cells in another tissue. And now that microRNAs as a phenomenon throughout all of biology have been recognized, everyone is trying to study whether you can find a fingerprint of microRNAs that will help you diagnose diseases, just the way we've been using that same fingerprinting or gene microarray technology to study full-length gene expression. And in several cancers, for example, as I think Dr. Petty said this morning, the fingerprint of microRNAs is actually a better descriptor both of diagnosis and of prognosis than the gene array of full-length messages.